be here with all of you, and we would like to show you what we studied about living things' vital functions in our high school.
Today, we have got a lot of responsibility because we are representing all the students of second year of ESO at INS Joan Oró in Lleida. First, we would like to introduce ourselves. We are Roger, Blanca, Isabel, Roc, and me, Blau.
In our high school, we work by doing projects. We do 2 projects each term, and we work on groups of 3 or 4. We feel as the protagonists because we have to search for information and we have to take the decisions. At the end of each project, we have to present our results to the city, to our parents, or to the high school.
Here you can see all that Roc explained in this video. Well, one of these projects was called Birthday Cake, and the main issue was cells and vital functions. In this project, we had to investigate Saccharomyces cerevisiae, that is a type of yeast, and discover if it's a living thing or not. So today, we are going to explain you our results.
We studied the main characteristics of living things. We discovered that they are all made up of cells and they all have 3 vital functions. Then we look if yeast had these characteristics or not. We carry out an experiment to see if yeast is made up of organic molecules, cells, and we discovered that yeast cells are between 3 and 4 centimeters of diameter.
The experiment is known as the elephant's toothpaste. To carry out this experiment, the main materials were hydrogen peroxide, soap, yeast, and a coloring.
First of all, we mix the hydrogen peroxide, the soap, and also the red coloring. And then we poured the yeast dissolved in water in the first mixture. We wait for some minutes, and we see that the mixture gets expanded and bubbly.
The result of this experiment is due to an exothermic reaction. This reaction happens because yeast contains an enzyme called catalase. This enzyme catalase breaks the hydrogen peroxide in water and oxygen. And this oxygen go out very quickly, and it forms this type of bubbles that we can say that is toothpaste of elephants.
The yeast is a biological catalyst because it turns an enzyme called catalase. The catalase is an organic molecule, a protein that yeast has synthesized.
On the other hand, we also carried out some experiments to see if yeast has the 3 vital functions. Here you can see the 2 posters. The first one is about nutrition and the second one about interaction. We didn't carry out any poster for reproduction, but then we talk about it. In the first poster, it talks about the nutrition. It talks about the experiment that is called yeast balloons. It's called like that because we use yeast to blow up a balloon. This experiment is used to see what does yeast eat.
Yeast are unicellular microorganisms in the fungi kingdom. They are facultative and aerobe, which means that they can breathe or ferment depending upon environmental conditions. When oxygen is available, respirations take place, aerobic respiration, and when oxygen is not available, fermentation occurs, anaerobic respirations. Both processes require sugar to produce energy. So that means that yeast needs food, in this case sugar, to produce energy. Here you can see the cellular respiration, that is the process that cells use to transfer energy from the organic molecules in food to ATP, energy, when oxygen is available. And then you can see the chemical reaction of fermentations, which produces ethanol and carbon dioxide as metabolic waste products.
Here you can see the materials that we needed. The most important ones were sugar, yeast, and also balloons. And well, we do we did two mixtures. The first one with yeast and water, and the second one with yeast, sugar, and water. In both cases, the water was at 35 degrees, and then we attach a balloon over the mouth of each flask, and we wait for some minutes to see if sugar fits yeast.
Well, this is a process, as Blau have just explained. Here you can see that the yellow balloon, it only contains sugar and water. Yeast and water. So it didn't blow up. But however, in the blue balloon blew up because it contained yeast, sugar and water. So the sugar reacts as a foodstuff, and for that reason it blew up.
The yeast uses sugar and warm water to grow. Warm water provides heat to the yeast activity and accelerates it. As yeast grows, it expands and gets bubbly. Yeast gives off carbon dioxide, and this gas inflates the balloon. And this is the reaction. The yeast also produces ethanol. Ethanol fermentation is used to produce beverage and also to make fuel. Yeast is nourished by sugar. It means that yeast uses sugar to grow. We saw that we can blow up a balloon with yeast. So we have another clue that yeast is alive, but we still have to study if it interacts and reproduces.
The other experiment is called the effect of temperature on yeast activity, and it's used to see if yeast interacts. The materials we need to do these experiments are yeast, sugar, and very important, water. The water has to be at 20, 30, 40, 50, and 60 degrees.
In this experiment, we prepared 5 different mixtures with yeast and sugar, each one at a different temperature, and then we measured the length of the foam of each graduate cylinder for 25 minutes. Finally, we compared all the results and we made a graph.
We can measure the rate of fermentation in yeast by measuring the amount of carbon dioxide it produces. Carbon dioxide production can be determined by the length of the foam on the top of the yeast solution, which catches carbon dioxide. The length of the layer of the foam we measured in millimeters because it's very accurate. And in these photos you can see the different temperatures after 10 minutes and the foam on the top of the yeast solution. And it does like a wave. And Isabel, please explain.
Yes. Yeast activity is higher between 30 and 40 degrees. And it's higher at between 30 and 40 degrees than in 20, 50 or 60 degrees. Below 20 degrees, yeast isn't in its comfort temperature because it's too cold. And at 60 degrees, yeast dies because it's too hot. So this graph shows how yeast activity changes depending on the temperature.
As you can see, the temperature is shown by different colors, and the foam length is measured in millimeters in the vertical axis, and the time is measured in the horizontal axis in minutes.
The highest amount of foam that yeast produced was at a temperature of 40 degrees after 25 minutes. It produced 280 millimeters of foam, and the lowest amount of foam that yeast produced was at a temperature of 60 degrees after 5 minutes. It produced 1 millimeter of foam.
Many factors go to justify the results. To begin, if the water was too cold at 20 degrees, the yeast cells start moving slower, causing less reaction, so the cellular respiration rate goes slowly and decrease. If the water was too warm, at 50 or 60 degrees, and the enzymes of yeast denature. When the enzymes of yeast denature, the cellular respiration rate stops and yeast die.
Reproduction is sexual, has an asexual type of reproduction called budding. From one cell of the yeast, it grows another smaller, and when it develops, it falls off and a new yeast cell is produced. We can see that reproduction happened in all our experiments, because if not, we couldn't obtain these results.
And this is the end of our presentation. Thanks for your attention and for giving us this opportunity to participate in this conference.